Okay, hi everyone, welcome to the session. So today I want to provide some information on targeting viral infections therapeutically. So you may have had some information from other modules on how to target microbial infections therapeutically, and that will have included some information on viral infections. But what I want to do now is focus in on some of the issues that are particularly relevant to viral infections, kind of look at some of the common viral infections that we need to think about from a therapeutic perspective and think of the issues that are dealt with. Obviously, we're not clinicians, and that's not the angle we're going to be taking. We're going to be taking a research scientist perspective and think about some of the issues there and some of the work that needs to be done to deal with these viral infections in a more effective way. And again, we're going to be taking a rounded perspective. We're not just thinking about the specific antivirals that need to be used in a specific viral infection. We're thinking about the context. We're thinking about the socioeconomic factors, all these things that really impact the ability of scientists to control viral infections in a real world environment. So firstly, we do want to talk about antivirals a little bit just because we want to establish what they are. And I know we've discussed this at points in the past, so I'm not going to go into a huge amount of detail here. I'm just going to establish some of the key concepts and make sure we're completely clear what we're talking about when we're referring to antivirals. What we're essentially referring to with the term antiviral is drugs that inhibit this viral replication cycle here. So as we've discussed, this is the generic viral replication cycle that all viruses undergo. The details of how they undergo this cycle differ. However, the key stages are always there. You always have viral attachment and entry, and then replication, and then assembly of new viral particles, and then release of these new viral particles. So these are the generic stages of a viral replication cycle. And again, what antiviral drugs do is they inhibit specific stages of this replication cycle. Just a little bit of more information on antivirals here. And again, we're talking about general principles around antivirals rather than specific antivirals here. I'm just emphasizing the point that antivirals and antibiotics are obviously different, and also emphasizing the point that antivirals are particularly difficult to develop. They have some very specific issues that kind of present a barrier to the development of antivirals. One of the particular barriers is the fact that viruses are intracellular parasites. So when a virus is infecting a cell, as we know, it enters into the cell. It's an intracellular parasite and it manipulates the host cell processes. It therefore makes it quite difficult to target drugs that specifically inhibit viral replication without also inhibiting host cell replication, because the viruses are using host cell machinery. So toxicity, also known as side effects, are issues with antiviral drugs, and some quite impressive lengths have been gone to in some cases to develop antiviral drugs that don't induce toxicity, so find a way around this problem essentially. The issue of animal models is also present. So when developing antiviral drugs, you want to develop them in such a way that you are testing them in animals before you're taking them to humans. Obviously, it's just a safety thing. It's just good practice. It's the way things are done. We need to know they're safe in animals before they're safe. Before we discover if they're safe in humans. However, the issue is, for a lot of viral infections, there aren't any animal models. So in a perfect situation, if you want to test an antiviral to be used in the case of viral disease A, you need an animal model that can become infected with this virus, so that can develop viral disease A, and then you attempt to treat this viral infection, this viral disease, with the antiviral, and you see if it works. And that's the first stage of your development. You're testing how it works in animals. If you don't have a model for that disease, so if you don't have an animal that can develop the disease you're looking at, then you're limited in how you can test the antiviral. You can give it to healthy animals, and that's fine, but it's not good enough. Generally, you want to be testing the actual model of disease, and if the model of disease doesn't exist, as is the case for a lot of viral infections, then you're in a bit of a difficult situation. Just a bit of a technical distinction here. We do want to be aware of the difference between antivirals and antiretrovirals. And the principle there is antivirals target viral infections, antiretrovirals target retroviral infections. So antiretrovirals are specifically used against retroviruses. However, some antiretrovirals can also be used against similar viruses that aren't retroviruses, but they engage in similar processes within the host cell. And the virus I'm thinking of there is hepatitis B virus. If you remember when we were looking at viral replication, there were similarities between hepatitis B virus and the retroviruses, and this is why you can use some antiretrovirals not just against retroviruses, but also against hepatitis B virus. And summarizing the bottom, all retrovirals are antivirals, but not all antivirals are antiretrovirals. So antiretrovirals are a group of antivirals. Okay, so again, sorry if you've seen the slides before, sorry, hopefully I went through them nice and quickly. It's just to make sure the points are established. What I want to do now is go into a little bit more detail on some things. So the distinctions here are really, really important. So we're essentially looking at antiretrovirals here, and we're looking at reverse transcriptase inhibitors. You remember reverse transcriptase is an enzyme that is used, it's the principal enzyme that's used in reverse transcription. So it mediates reverse transcription of the retroviral genomes. We spent some time focusing on this process of reverse transcription. It's going from an RNA genome to a DNA genome, and it's a prerequisite for the integration of that DNA genome into the host cell genome. Now, this is a really, really attractive target, therapeutically speaking. And the reason for that is that, as we discussed, reverse transcription never happens in a host cell. It's a completely foreign process. Because it's a completely foreign process, we can target that specific process without impacting healthy cells and without impacting the cell as a whole. So generally speaking, as we've said, viruses will co-opt and manipulate host cell processes. So they will utilize host cell enzymes and things like that. Here we have an example of a virus not doing that, of a virus using its own enzyme, which does happen. It does happen. It's not particularly rare, but when it does happen, what we want to do is target that enzyme with drugs because it presents a unique target, a target that isn't present in the cell unless the virus is there. So whilst generally speaking, viruses are using host cell processes, whenever they do use their own processes, it presents a useful therapeutic target to us. And that's exactly what happens with reverse transcription, mediated by this enzyme, reverse transcriptase. Now, when we're talking about retroviruses in terms of human health, the most important retrovirus is HIV. So when we were treating antiretroviral infections, when we're treating HIV infection with antiretroviral drugs, almost always one of the drugs being given will be a reverse transcriptase inhibitor. Now, you'll probably remember the therapeutic regime to treat and control HIV is HART, which is a combination therapy. So it's 3 different antiretrovirals that are given. Almost always, at least one of them will be targeting reverse transcriptase. There'll usually be a mix, so could be protease inhibitors as well and fusion inhibitors, but one of the 3 at least will almost always be a reverse transcriptase inhibitor. One of the reasons for that is because it's a process that's unique to the virus, so it's limiting side effects and toxicity. The other reason is that it's a fundamentally important process. It's one of the core processes in the middle of the viral replication cycle. So it's a really, really attractive target. So hopefully that makes sense. Now we have two distinctions at the bottom. We have the NRTIs and the NNRTIs, the nucleoside reverse transcriptase inhibitors and the non-nucleoside reverse transcriptase inhibitors. And what we're going to do now is discuss those distinctions. So we've identified reverse transcription as a really attractive therapeutic target if we want to inhibit the retroviral replication cycle. And again, reverse transcription is the process that's defined on the side of the screen here. So we're going from an RNA genome that is single-stranded, so the light blue, to the darker blue double-stranded DNA genome. And again, this is what reverse transcription is, and it's mediated by the viral enzyme reverse transcriptase. This double-stranded DNA genome that's produced can then be integrated into the host cell genome, where it forms the provirus. If any of this isn't familiar, then if you can go back a few lectures, we did talk about this at length. We really went into the details of this process. These reverse transcriptase inhibitors are drugs that are disrupting this process, so they're preventing this from occurring. We can actually split the reverse transcriptase inhibitors into two groups dependent upon their mechanism of action. So there are two principal ways in which we can target this process of reverse transcription. One is to target reverse transcriptase directly, so we can take drugs, we can develop drugs that specifically target reverse transcriptase and prevent it from working, usually by binding directly to the drug, to the protein. So the drug, the antiviral, will bind to and inhibit reverse transcriptase, and therefore will prevent reverse transcriptase from fulfilling its job, will prevent it from mediating this process of reverse transcription. These are the non-nucleoside reverse transcriptase inhibitors, also referred to as the NNRTIs. If you're reading literature around retroviruses and around antiretrovirals, you'll see this term thrown around, NNRTIs. That's exactly what it means. It means non-nucleoside reverse transcriptase inhibitors. These are antiretroviral drugs that directly inhibit reverse transcriptase. So what does this terminology actually mean? What does non-nucleoside mean? The reason why we use that terminology is because it's distinguishing these antiretrovirals from the second group of reverse transcriptase inhibitors, which we're going to define now. And these are the nucleoside analog reverse transcriptase inhibitors. So both these groups are reverse transcriptase inhibitors. That's why they end in RTIs. But this group we're looking at now are nucleoside analog reverse transcriptase inhibitors. These are NRTIs. So what does this term mean, nucleoside analog? So essentially, these things look like nucleosides. They're very, very similar to nucleosides. However, they're slightly different. That's why we use the term analog. When these nucleosides are, or nucleoside analogs, enter into the cell, so an individual is being treated with these drugs, the nucleoside analogs will be processed into nucleotide analogs just by they'll be phosphorylated just by normal cellular proteins, by kinases that do these phosphorylations. So then in the cell, we have nucleotide analogs. These nucleotide analogs are similar enough to nucleotides, so they look enough like nucleotides and they have properties that are similar enough to nucleotides to be included in new chains of DNA that are being produced by reverse transcriptase. Okay, again, that's what's happening when this DNA is being produced, because remember, reverse transcriptase is a polymerase. It's polymerizing nucleotides into DNA. If this drug is present in the cell, then these nucleotide analogs will be incorporated into the DNA. And this is how the drug works. It is, by virtue of being an analog, it is similar enough to be incorporated into the DNA. However, it is different enough that it causes chain termination. So reverse transcriptase is polymerizing nucleotides to produce DNA, and it's using RNA as a template, right? It's adding nucleotide, nucleotide, nucleotide. However, once it adds an NRTI, so a nucleoside analog reverse transcriptase inhibitor, um, it causes chain termination, it aborts the process, and the reason is that this drug, once it's processed into a nucleotide, is similar enough to a normal nucleotide to be recognized by reverse transcriptase and be incorporated, but it's different enough, so it has these specific differences to normal nucleotides that mean additional nucleotides can't be added onto the end. So it's a really clever mechanism, and it essentially means that once this nucleotide analog is in the cell, it causes abortion of this whole process. It means reverse transcriptase can't continue because these nucleotide analogs are included in the place of normal nucleotides, and by being included, they're disrupting the whole process. So hopefully it's clear that these that we've just talked about, the NRTIs and also the NNRTIs, so the non-nucleoside reverse transcriptase inhibitors, are both capable of disrupting this process of reverse transcription, but they do so by slightly different mechanisms. So the NNRTIs by binding to and inhibiting reverse transcriptase directly, the NRTIs on the other hand by being incorporated into the nascent DNA. Nascent means forming, so not yet fully formed. So the nascent DNA, we sometimes use it to refer to DNA as it's being formed. And yeah, so the NRTIs cause disruption of reverse transcription by being incorporated into the nascent DNA and causing disruption of the process that way. Okay, so that's how the reverse transcriptase inhibitors work. The other groups of drugs, the protease inhibitors, the fusion inhibitors, the inhibitors of release, the inhibitors of attachment, we've talked about, or you should have talked about these in other modules. And we have talked about them a little bit in this module, so I'm not going to labour that point and go over it again. I just wanted to provide that technical information on those specific drugs. Now we'll think about HIV as a persistent virus, so as a latent infection. So this is a long-term infection, and what I want to do here is just to emphasize the fact that HIV is not unique amongst the viruses, but quite unusual because it can go into this persistent state. So it has this long-term latent chronic state in which it goes silent. And again, the reason for this is because it is integrated into the genome, because even if you're treating an infection with antiretrovirals, you're just suppressing replication. You're not getting rid of those latent reservoirs that exist. So as we said, despite HIV being a chronic infection, we can control it with the drugs we were just talking about, with the antiretroviral drugs. We have to use a combination. The first drug that was used to treat HIV, so the first antiretroviral developed, was azidothymidine that you may have heard of. It's actually an NRTI, so it's a nucleoside reverse transcriptase inhibitor. So one of those groups we were just looking at, and it's really effective at first. However, very, very quickly, HIV developed resistance. It developed, it evolved resistance to the drug, and the drug wasn't very effective anymore. And that's when it was determined that a combination therapy had to be used, which eventually became highly active antiretroviral therapy. First, it was just ART, so antiretroviral therapy. Then it was highly active antiretroviral therapy. And again, one of the issues with viral infections, and you'll see this all over the place, is they have a very, very high mutation rate. When we're looking back at the lectures discussing the replication methods, we did touch on why these viruses have such high mutation rates. And just on the surface, it makes sense because they have such a high rate of production of progeny. Each cell that's infected with the virus can potentially be pumping out tens of thousands of new virus particles. So there's so much opportunity there for mutations to occur at such a high rate of progeny production, such a high reproductive rate. It's really not surprising that we see drug resistance very quickly in these viruses. However, if we're using a combination of drugs to treat viral infections, we reduce the possibility of resistant strains emerging, because any resistant strain would need to have resistance to in the case of heart, the 3 antiretrovirals being used simultaneously, and it's very unlikely for that to happen. Okay, so one strategy that's been used to deal with these latent viruses is shock and kill. And there's been a lot of interest around this in HIV, not so much around some of the other latent viruses. So there doesn't appear to be any papers on herpes viruses looking at shock and kill. Maybe you can find some, but I couldn't find any at all. Well, apart from a few from quite a while back discussing the possibility. For HIV, however, there was a lot of interest in this area, and the idea was if we can induce latently infected cells to start producing HIV, that will allow us to target them using antiretroviral drugs and other therapeutic interventions. So the idea is you're shocking these latently infected cells into action, you're shocking them into expressing the HIV genome, and then you're killing them with drugs. Again, the term is shock and kill. You're essentially forcing these cells to expose themselves, forcing the reservoirs to demonstrate the fact that they're infected, and then they can be targeted. There's a lot of papers on the side here that show some promise. They're fairly recent. However, there's a paper in the bottom left here, which is the most recent paper that you can come across, saying they don't really seem to work very well at all. This is quite a strong statement, actually. I know it's saying unconvincing results, but the unconvincing result, it's not an endorsement of the therapies. Basically, it says the clinical trials haven't been successful in proving that these shock and kill strategies can be effective. Now, the papers are up and down, so some say it's okay, some say it's not, but it's definitely not a resounding success. There's no reason why these shock and kill strategies couldn't work in the future. It's not looking great at the minute. Again, the principle of using these latency reversing agents, and again, these are the drugs that will cause the cells to start expressing the HIV genome, so you're reversing latency, and then you're treating with antiretrovirals, then you're targeting infection itself. The principle is sound, so it could just be that it hasn't been developed to a point where it's efficacious yet. Okay, so there have been some perspectives on antivirals that hopefully were novel. What we want to do now is move towards vaccination and start thinking about vaccines. So again, I know you've discussed what vaccines are. What we want to do now is look specifically at some vaccines. Okay, so just to start off with some general points. These are the distinctions between antiviral drugs and vaccinations. So as we know, antivirals are used to treat infections, whereas generally vaccines are used to prevent infections. This isn't absolutely true, as we're going to see in a few slides, but these are general principles. Again, antiviral drugs are targeting the viral replication cycle. Vaccines are priming the immune system. They're immunizing you so that you can respond appropriately to exposure to a particular infectious agent in the future. Vaccines are generally considered the gold standard, and the principle there is there's barriers that are overcome inherently by vaccination. So with drug usage, you have the barriers of maintaining usage, and there's things like pill fatigue. As we've discussed, there's often resistance to drugs, so resistance can develop in the microorganism, or in this case in the virus. So these are real issues, and these matter. And if you have successful vaccination, you avoid these issues, you overcome those barriers. You don't have to worry about pill fatigue, you don't have to worry about ongoing cost, you don't have to worry about ongoing access to the drugs. So these are just some of the general evaluative points that you need to be aware of. So you not only need to know the difference between vaccines and drugs, but you need to understand this difference in detail. You need to understand the strengths and the benefits of each. And there are strengths and benefits of each, but there's also weaknesses of each. So even though we consider vaccines the gold standard, they're not perfect. one way in which they're not perfect is compliance issues. So I'm sure we're aware of anti-vaxxers, so people who are disinclined from becoming vaccinated for various reasons. There's also issues like cold chains, which I think we'll talk about later. So the idea that many vaccines need to be stored at a particular temperature. This is an issue when it comes to distribution, particularly if distribution is in portions of the world where they they don't necessarily have the infrastructure to make this easy. So it can be quite difficult to transport vaccines to rural populations below a certain temperature just because the infrastructure isn't there. And these evaluative points are outlined here with a few more as well. Obviously with antiviral drugs, generally we need diagnosis first. You need to know what drugs to give someone, so you need to know what viral infection they have. This isn't really the case with vaccination. With vaccination, you're just vaccinating the population, so there's no diagnosis necessary. You're just vaccinating as many people who will comply, essentially. But of course, this compliance in and of itself is an issue. Another issue with vaccination is that even if you want to vaccinate everyone, you can't do that because some people just can't be vaccinated. So for health reasons, this is kind of a twofold problem. Some people, it's not safe to vaccinate them because some vaccines are, as we've discussed, they're attenuated, so they're just weakened forms of the virus or the microorganism. And in a healthy person, that's absolutely fine. The attenuated form won't cause disease because it's weak. But if you have an immunocompromised individual, so an individual who has an increased susceptibility to infection, then it's potentially dangerous to give them an attenuated vaccine because they could become harmed. They could have an active pathological infection caused by this attenuated virus. So obviously there's an issue there, and as I said, this issue is twofold. The other issue is if an individual is immunocompromised, so they don't have a fully functioning immune system, then they in all likelihood won't respond to the vaccine anyway. So even if you vaccinate them, even if the vaccine isn't an attenuated vaccine, even if it's a killed vaccine or a toxoid vaccine where it's just it's a protein taken from the virus, it it won't stimulate an immune response because they don't have a functioning immune system. Again, they're not immunocompetent, they're immunosuppressed. So if you have individuals who are in this immunocompromised state, it could be very difficult to induce an immune response. So even if you vaccinate them, you can give them multiple booster shots, and just what you'll see is they won't respond to it, so they won't be producing antibodies, and therefore it's almost a waste of time, isn't it? So you can't successfully immunize them with vaccination. And again, I'm not making a huge point here saying one is better than the other, saying vaccines are better than drugs or drugs are better than vaccines. I'm just saying there's a discussion to be had. There's contextual issues as well. So in one context, vaccination can be preferable, but in another context, drug-based interventions can be preferable. Generally speaking, as a community, scientists tend to consider vaccination as the gold standard, and the success we have had in eliminating specific infectious diseases, namely smallpox, was done on the back of vaccination. So not to diminish the importance of antiviral drugs, but vaccination is considered the gold standard generally. And there's just a bit more information here on what vaccines are. I'm sure you'll already know this, it's just so the information's there. And again, the principle here is the way vaccines are working is they are priming your body to have an immune response to a particular microorganism, in this case virus, in the future, and that's it. So if an individual becomes vaccinated, hopefully they are then immunized and primed to have an immune response to the particular virus they've been vaccinating against. There are some stipulations as to what makes an effective vaccine. Firstly, it must be safe. So obviously it can't cause harm or it can't be used. Secondly, it must be efficacious, so it must actually provide protection against a particular infectious agent. The response must be long lasting. So if you have a vaccine that only provides a protective response for a short period of time, so after a period of time, an individual's immune system can no longer be considered prime, it's not a great vaccine. It can still be good and it can still be useful, but it's not ideal. Ideally, we want a long-term response, potentially a lifelong response, because some of these vaccines, if an individual becomes vaccinated, particularly with booster shots, they have lifelong protection. And this is really crucial here. The efficacy of the vaccine must be significant enough to counteract any behavioral changes. And this is something we've touched on in the past, I think. This idea that if you vaccinate an individual, they will assume they are protected and they will then potentially alter their behavior, and this alteration in behavior could increase their exposure to a particular infectious agent. Now, if the vaccine only provides a low level of protection, then their alteration in behavior could actually result in them having an increased likelihood in becoming infected. So the vaccination indirectly has actually increased their likelihood of becoming infected, not because it's had any biological impact that has increased their susceptibility, but because it has induced a behavioral change that has increased their likelihood of exposure. And this is really, really important. So when you talk about, or when you hear about us needing a particular level of efficacy for a vaccine, that's it's kind of twofold. One is because it's trying to induce herd immunity. The other is because we need to counteract behavioral changes. There have been vaccines that have been developed against infectious agents in the past that have been, say, 30 or 40% effective, that have been essentially discarded, that haven't been developed any further. And the reason is that that is just not good enough. On the surface, we can say, oh, 30 or 40% protection, that's pretty good. We can reduce 30 or 40% of cases. However, that's not actually how it works, because people change their behavior. So we just want to be aware of that as well. This final point is to do with the practicality of developing these vaccines. So an ideal vaccine is able to be produced in a mass quantity, so at a production rate that enables it to be actually useful for populations. Again, when we're talking about vaccination, we're talking about treating, or we're talking about vaccinating large numbers of people. This is in contrast to drugs generally, because what we're doing with drugs is we're targeting specifically individuals who have been diagnosed with a particular infectious disease. Generally, this is what we're doing with drugs. Vaccination is different. Vaccination is a blanket approach, and we therefore need the practical ability to support this approach by producing enough of the vaccine to vaccinate as many people as possible. Now, when we're referring to vaccines, we tend to be thinking about active vaccines, and it's generally almost always what we're talking about. However, we also need to be aware that passive vaccines exist. Passive vaccines, they don't fit the traditional criteria of what a vaccine is. However, we do define them as passive vaccines, and it does kind of make sense. The principle is that an individual is not having an active immune response, so the vaccine isn't immunizing them directly through their own immune system, but it is providing them with protection by inducing them with the products of another individual's immune response. And what I mean by that is often passive vaccination involves the injection of antibodies or antibody complexes. It's comparable to, so that there's two types of passive vaccination. What I just referred to as artificial passive vaccination, where it's used therapeutically, where you can take an individual who's been exposed to a particular infectious agent, you can isolate their antibodies or just isolate their blood plasma and process it appropriately, and then use that product to provide another individual with protection. That's an artificial passive vaccine, and it's comparable to a natural passive vaccine, which is the best example of a natural passive vaccine is the passage of antibodies from a mother to the baby. So this maternal protection that an individual derives. So these antibodies are passed from the mother down to the baby, and the baby is therefore experiencing protection against particular infectious agents without ever actually having had an immune response to those infectious agents. So it's deriving the protection indirectly. It's passive protection, and that's what we tend to consider these passive vaccines. And you can hopefully see from this data here, the papers in the bottom, in the notes of the slide, the ZMapp improves current standard of care. So if you have current standard of care with ZMapp, you see higher patient survival, not by a huge amount, but clearly there's a difference there. Again, it's just the paper, it doesn't meet their criteria for efficacy. It's also quite useful for us to know about these attenuated vaccines. So I discussed earlier in this lecture what attenuated vaccines are. Just to clarify that, attenuated vaccines are composed of versions of the virus or the microorganism you're immunizing against that have been sufficiently weakened that they can't cause pathology. So it's a version of the virus that is often replication competent, so usually it can replicate, however it can't cause disease, so it's been genetically modified in some way that it is not able to actually cause pathology. This is potentially an issue in immunocompromised individuals because, as we've said, an attenuated virus that can cause no pathology in a healthy individual could hypothetically cause pathology in an immunocompromised individual. So that's something we have to be very aware of and we have to consider. However, in healthy individuals, attenuated vaccines, there's no issue at all. There really shouldn't be an issue in immunocompromised individuals either, because there's so many safety levels to go through. But the worry is there, the potential is there. So it's something that is worried about. There's two examples on the right. One is of the Savin poliovirus vaccine. Savin poliovirus vaccine, this is an attenuated vaccine that just has a 5' mutation in the non-coding region. So this is a region in the 5' end of the genome. And this mutation means viral replication is impaired, and just by impairing this viral replication, the virus loses the ability to cause pathology essentially. However, and this is crucial, it maintains the ability to induce an immune response, because that's how vaccines work, right? So these attenuated vaccines, they retain the ability to induce an immune response, but they lose the ability to cause pathology. The herpes virus vaccine that is currently in development. I'll put the paper in the notes in the notes at the bottom of the slide, so you'll be able to see it there. We can call it we can almost call it a hypothetical vaccine, although it is in development and it's showing a little bit of promise. What essentially occurs is there's a mutation in the viral helicase, which is a specific viral enzyme carried by the herpes virus, and this mutation again impairs the ability of the virus to cause disease, but doesn't reduce the ability of the virus to cause an immune response. And I'll put the paper there. I think it's quite a preliminary paper, so there's no necessarily any reason to believe this is going to go anywhere. It's not close to being used as a vaccine, for example, but the principle is there. We can see the principle of these attempts to develop attenuated vaccines, and the Savin poliovirus vaccine, this actually does work. This is approved. So these attenuated vaccines are viable. They're still being developed against various viral infections. Killed vaccines, this is another type of vaccine you should be aware of. So some vaccines are just killed forms of the virus or the microorganism. Often the virus will be fixed in formalin, so it will be fixed in a way that it retains its structure, so therefore it's still able to induce an immune response. If you kill the virus in such a way that it just breaks apart, then that's not very useful, is it? It's potentially not going to induce an immune response. However, if you fix it in formalin, then hopefully you're still able to induce an immune response, despite the fact that the virus is not capable of causing disease, because it's fixed. It's not replication. It's not replication competent at all. It's fixed in formalin again, usually. The potential for disaster exists here and has been realized in the past, something called the Cutter incident, where a poliovirus vaccine, and this is a killed vaccine, was not sufficiently inactivated, so it was not sufficiently killed, and a large number of individuals were infected with what amounted to infectious poliovirus. Obviously a huge issue. There was a huge human cost to that, and a lot of individuals died. Instances like this are unfortunate. I mean, they don't impact the validity of vaccines overall. It's just procedural errors, so it's just mistakes that have been made along the way. One of the unfortunate aspects of this is it gives people who are disinclined from vaccination justification. It gives them firepower to argue against vaccination, and there are absolutely valid criticisms of vaccination that exist. However, instances like this, it's more to do with the procedures being followed, it's more to do with mistakes being made rather than the principle of vaccination being at fault. So the idea of using inactivated virus particles as a vaccine is absolutely fine, and it's absolutely safe if it's done properly. And what happened here was it wasn't done properly, and the virus wasn't inactivated, and a large number of individuals were unfortunately infected. So now we want to discuss the DNA and RNA vaccines a little bit. These are interesting. These are the kind of, we can think of them as quite modern vaccines because they're using quite advanced technology and things like that. The idea is that if you can get a cell to uptake DNA or RNA and then express that DNA or RNA into protein, so if it's DNA, the term is express. If it's RNA, I guess the term is translate, because it's just directly translating the RNA into protein, right? Then you can get cells to produce specific proteins, and if these proteins are proteins that induce an immune response, then you are therefore inducing vaccination potentially, and that's the principle behind DNA and RNA vaccines. You're introducing genetic material into host cells that these host cells are then using to produce protein, and this protein is then inducing an immune response. So it's just a different way of vaccinating, just a different approach to directly vaccinating with the microorganism or with protein. You're vaccinating with the DNA or RNA, and then the host cells are producing the protein themselves. This is foreign protein, of course, because the DNA or RNA you are using encodes usually the viral protein or the microbial protein. That's the principle, because this is what's going to then induce the immune response. And these papers that are kind of appearing on the screen here, just some reading that you can potentially do. These are mostly from the 90s, so these are quite early papers establishing the principles of DNA vaccination. So they're quite interesting and they're quite useful in that regard. RNA vaccines principle is the same, just slightly different because it's RNA that is being introduced in the host cells, and then this is RNA that is being translated into protein rather than DNA being fully expressed and then ultimately resulting in the production of protein. RNA vaccines are interesting at the minute because they're relevant to COVID. So there's COVID RNA vaccines that are being developed, one of which is mRNA 1273. So I have a quote here from a paper that's very recently been published, describing how the candidate mRNA 1273 encodes the SARS coronavirus 2 spike protein. Now this spike protein, if you're looking into vaccines against SARS coronavirus 2, you'll see this spike protein come up again and again and again. This is the portion of the virus that seems to be able to induce a protective immune response, so seems to be very useful as part of a vaccination. So a lot of the different vaccines are utilizing this spike protein. And this paper describes that this particular mRNA, mRNA 1273, is translated into the spike protein, and this mRNA induced an anti-SARS coronavirus 2 immune response in all participants. There were no trial limiting safety concerns, and these findings therefore support further development of this vaccine. So this is one of the vaccines that is looking potentially promising for COVID-19. So obviously we have a lot of vaccines that are being developed at the minute, and they all have, not all, but a number of them show some promise. And are at different stages of development, and it's actually likely going to be we end up in a situation where we have multiple vaccines that can be used. That's actually not a bad thing. Different individuals will respond differently to different vaccinations, and there's also issues with supply. So again, the kind of practical issues we talked about, like how do you produce as many copies of the vaccine as you need to vaccinate a whole population, never mind populations around the world. So having choices of different vaccines is never a bad thing, really, particularly not in the context of a global pandemic. You can also see a paper on the left here, which is discussing a particular mRNA vaccine being used to prevent HSV, so herpes simplex virus infection. This is also a recent paper, so this is 2020, so this is really interesting. It seems to be really effective in the paper, but this is in mice, so these are animal studies, this is an animal study, I should say. It's not 100% confirmation that it will be anywhere near this effective in humans, or it will be effective at all in humans. It's just an animal study. It is what it is. However, it is promising, so it's indicative that the mRNA vaccine for herpes simplex viruses may in fact work. This is another mRNA vaccine being developed in attempts to prevent SARS coronavirus 2. This also looks promising, being developed by a different company. The links and stuff will all be made available if you want to read up on these. Again, the principle is the same. The mRNA is taken up by the host cells. The mRNA is translated into protein. This protein induces an immune response, and then that immune response is protective against infection. This is the principle. It appears to be working fairly well, though we have to remember these two studies of the mRNA vaccines to protect against SARS coronavirus 2. They have their limitations, and they're just single studies at the end of the day. I think this one is actually an interim analysis, or I think if you read up on it's in the sources. One of these studies is just an interim analysis, so it's not actually a finalized clinical study. It's just analysis of the data halfway through a clinical trial, just to see how things are going. So obviously there's limitations there. Okay, so that's vaccination. So hopefully we've gone into a little bit more depth on antivirals and vaccination, and you feel like you have a bit of a deeper understanding of what they are, and a bit more of an understanding of the issues surrounding them and things like that. Now we want to talk about self-limiting infections. Now, self-limiting infections are a huge part of virology. A lot of viral infections are self-limiting, so we need to understand what we mean by the term. Essentially, when a viral infection is occurring, there's a few outcomes that could potentially be arrived at. So it's perfectly possible, and in a lot of cases of viral infections, the immune system is able to clear infection entirely. So the immune system clears infection, and therefore we can consider it self-limiting. So the body is able to limit infection, the immune system is able to clear infection. Obviously, this isn't always the case, and sometimes infections are persistent, they are chronic, or they are latent, and we know that chronic, latent, persistent viral infections exist. There, it is desirable that we have therapeutic interventions to treat and cure the viral infection. Also, even infections that aren't necessarily inherently chronic or latent, there can be situations where the immune system is not able to clear infection. So there can be acute infections that overwhelm an individual's immune system. Again, this happens often. It's something that happens. In these cases, it's desirable that treatments are available and that there are therapeutic interventions that can treat the viral infections. Unfortunately, a lot of viral infections we don't actually have treatments for. That's just the way it is, or we have limited treatments for, so we have a low number of treatments for. So what we actually rely upon often when an individual has a viral infection are rather than curative treatments, so rather than antivirals that are actually targeting the viral infection, we rely upon symptomatic treatments. So just treatments that deal with the symptoms. So anti-inflammatories. Anti-inflammatories aren't actually treating the infection. They aren't targeting the viral replication cycle. They're just reducing inflammation associated with the infection. Nasal decongestants, they're just dealing with congestion. They're not actually dealing with the viral infection itself. Antipyretics, painkillers, anticonvulsants, these are all dealing with the symptoms rather than actually dealing with the infection directly. And that's fine. I mean, I'm not saying that's a bad thing. I'm just saying it would be desirable to be able to treat the infection directly. However, unfortunately, for a lot of viral infections, we don't actually have effective antivirals. Partly that's because, as we discussed at the start of the lecture, there are significant difficulties in developing antivirals. It's just the reality of the situation. Obviously, we do have some antivirals. We do have just over 100 antivirals. As we said, a lot of those are used to treat HIV, but a lot of them are also effective against other infections. So we have antivirals to treat hepatitis B virus. We have some to kind of help deal with the influenza and things like that. We have antivirals that can treat herpes virus infections. These are not, however, capable of curing these infections. As we know, herpes virus infections can go latent, but what they can do is limit infections. They can prevent replication and they can prevent the symptoms. And this is for all herpes virus infections. With herpes simplex virus infections, there's things like acyclovir, but there's also acyclovir and I think it's ganciclovir for other herpes virus infections, so more systemic herpes virus infections like cytomegalovirus, Epstein-Barr and things like that. So there are therapeutic options out there for some viruses. However, overall, there's a lot of viral infections that we can't really cure with antivirals. Again, many of these are self-limiting, so the immune system deals with it itself. It's able to clear infection. Often, this can be aided, or an individual can be helped with symptomatic treatments, so treatments that are going to deal with the symptoms, because preferentially we always want to be able to deal with the infection itself, to actually cure the infection, to clear the infection, or if we can't do that, to suppress the infection like we do with acyclovir and herpes simplex virus outbreaks, and like we do with HIV and antiretroviral infections. And antiretroviral treatments, we're not clearing the infection with antiretrovirals, but we are suppressing it. If we can't do that, if we don't have the antivirals, we tend to lean towards symptomatic treatments, so treatments that combat symptoms. So those are the self-limiting infections. Hopefully they've been defined effectively. Now what we want to do is fill out some of the context around all these infections a bit more effectively. So when we're talking about targeting viral infections therapeutically, which is what we're doing today, it's useful to talk about the direct interventions we can make, but we also need to talk about the context, and we have done that, and we do that quite frequently. But what we want to do is just dive a little bit deeper into the context and just think about some of the specific issues that we face when trying to design and implement therapeutic interventions against viral infections. So one significant issue that you're going to see crop up over and over again when you're doing your reading and when you're kind of studying around the module is the importance of socioeconomic factors. So the importance of poverty and everything that's wrapped into that. This is, and again, it's something that is inherently linked to infectious disease, and that is almost beyond the scope of infectious disease to deal with. I mean, at a certain point, we just have to deal with the situation as it is and mitigate the damage as best we can. Okay, so rural communities in less economically developed countries are going to struggle to get access to drugs very often. This is an example. That's the reality of the situation. What can we do about it? How can we mitigate that damage? What steps can we take to make sure those individuals can access the drugs? And we're not looking for societal solutions. We're not looking for these broad economic solutions that we can't control. Although it's absolutely fine to talk about them and consider them, but what we also want is practical, reasonable solutions that can benefit with almost immediate effect. So what can we do in the situation we're in now? In terms of poverty, generally what we're talking about is where can we direct the resources. So where can we find resources and how can these resources be directed into these communities? You also see a lot of issues alongside poverty. So when you have poverty, you tend to often have lower levels of education, and there's a knock-on effect of that. If we just think about this example we've got here in the slide, so the example here is referring to mosquito net fishing. Now this is a phenomenon where in rural communities in less economically developed regions, often sub-Saharan Africa, there's an issue where mosquito nets that are provided are not utilized properly. So the idea of the mosquito nets is they're placed over individuals while they sleep, and then the individuals aren't bitten by mosquitoes, and therefore they won't contract malaria. So mosquito nets should reduce the spread of malaria. It was determined they weren't actually working as well as they should have been working, and the reason was because of a phenomenon called mosquito net fishing, where individuals in these rural communities, instead of utilizing the mosquito nets properly, they were instead using them to fish in the rivers. And this paper argues, and it's been argued in many places, that this phenomenon is linked to poverty. So individuals who were engaging in this practice, they needed money, they needed food, and an immediate way for them to solve that problem was to use these mosquito nets to fish, and that was preferable to them than using the mosquito nets as they were intended, and to prevent this long-term potential of maybe they'll get malaria, maybe they won't. It's a difficult issue because on the surface, providing mosquito nets to individuals at high risk of malaria is a good solution to the problem. It's going to prevent the spread to malaria, but when you actually look into the communities at what's happening, it doesn't seem to be going to plan. One potential way to counteract this is education, but again, education is and that is a good solution, that if we can educate individuals on the importance of using the net properly, there's a good potential that will work, but there's issues around that as well. As we'll see in a few slides, there's issues with education, but also who's going to pay for this education? How is this education going to be provided? There's no perfect solution to these problems. All we can really do for now is identify what the problems are, try and understand the problems, and where appropriate, suggest potential solutions. So a potential solution here would be education, even though there is a significant amount of room for evaluation of that strategy itself. So what do we actually mean by education? How would this be implemented? Obviously, a kind of overall ideal world solution to this problem would be to bring these communities out of poverty. However, is there much value in suggesting that without a reasonable path for it to being done? It's just kind of a vague suggestion. Sure, it would be amazing if that could be done, but is it a reasonable target in the scope of preventing malaria? Probably not. It's just beyond the scope of research scientists, really. We see these facts played out again, these links played out again when we're looking at things like condom use, so other behavioural changes or behaviours that could reduce transmission risk of particular infectious diseases. And we see indicators of poverty, so low socioeconomic status associated with lower uptake of these measures, of these behavioural changes. And it's difficult to tie the exact specific causes down, but it often again seems to be linked to education. So if individuals who are impoverished or living in poverty tend to have lower education, tend to be less informed, so they tend to be in a position that is they're in a position that in which they're not well prepared to make these educated choices, to make these informed, reasonable, rational choices about their own health. If you add onto this the fact that individuals living in poverty have other immediate concerns, so they have immediate concerns: where are they going to get their food, how are they going to pay their bills, etc., etc. It doesn't put, these concerns don't put them in a very a good position in terms of long-term decision making about their health. When you're overly concerned with these short-term worries that are significant worries, you're not well positioned to think about long-term health decisions. And it's like with the mosquito net, isn't it? Individuals need food, they need money, so the short-term benefit of being able to fish outweighs the long-term benefit of preventing malaria. And again, if we tie in education as well, we're talking about rural communities that may not understand what malaria is. They don't necessarily understand what infectious disease is. They don't necessarily understand what vectors of disease are. They don't understand that mosquitoes can spread a disease, and if this net prevents the mosquito from biting you, it can, or taking a blood meal from you, it can prevent the disease malaria from being transmitted to you. These concepts are sometimes in these rural communities so alien that there's just it's really difficult to induce these behavioral changes. If we want individuals to change their behavior, we need to justify the behavioral change. And again, this comes with education. And we see this in Western countries as well. We see individuals are more likely to change their behavior if they understand why, if they understand the justification for the behavioral change, if they feel the behavioral change is justified, and then they'll be more likely to make that behavioral change. And just to reinforce that, we see similar things with Ebola as well. And again, this is reinforcing the education issue. So one of the significant barriers faced by healthcare workers trying to treat Ebola and prevent the spread of Ebola was just the very, very limited understanding of science that was present in these rural communities. Again, getting individuals or encouraging individuals to comply with scientific measures and interventions and approaches is very, very difficult when the individuals don't have an understanding of the science, and they're still relying upon, they'll still ascribe these cases of Ebola to evil spirits, witchcraft, sorcery. They'll still be visiting witch doctors over medical professionals, things like that. Really, really dangerous. And again, it's a really difficult thing to deal with without education. And there's this balance where it's not rational to there's no blame on these communities for mistrusting science. It's just these are foreign concepts, these are alien concepts. These individuals are living in real communities. They're not necessarily educated, so they're not in a position where they are likely to believe strangers who essentially swoop in. Often they'll associate the infectious disease with the strangers who swoop in because the events happen together, right? All of a sudden people are getting really ill with Ebola, and there's also a lot of strangers appearing in contamination coats, and they're swooping people off into quarantine centres. It's all quite intimidating. It's all quite strange if these individuals don't understand scientific principles. So it's really no surprise that they will often have low engagement with research scientists and medical professionals. And this is just reiterating that fact. The situation has been known to get so bad where there's been attacking of healthcare professionals, butchering them, and all sorts of really awful things going on. And really, again, I would argue all this comes from lack of education and subsequent lack of understanding. Lack of education and lack of understanding isn't, confined to less economically developed countries. It also occurs in Western countries, obviously, and it's important that we kind of recognise that. On the left here, I have an evaluation from the press release for one of the mRNA vaccines for COVID earlier, and it essentially says one of the challenges is related to the ultra low temperature that the vaccine must be stored at. It's an mRNA vaccine. RNA is unstable compared to protein, so it has to be stored at a very, very low temperature right up until administration. Now on the right, I just have a tweet that's been floating around, and it is what it is. Remember, it's just a tweet, but also if you can see quite a lot of people have viewed it. It's over 1,000 likes and over 800 retweets, and it says someone purporting to be a doctor, doc something. Any vaccine that needs to be shipped and stored at -80 degrees isn't a vaccine, it's a transfection agent kept alive so it can infect your cells and transfer genetic material. Don't let them fool you. And this is genetic manipulation of humans on a massive scale. Shut it down. So this is so there's a lot of problems with this statement. Obviously, it's not demonstrating a solid understanding of what an mRNA vaccine is. It reads as if it's designed to scare people and designed to influence people and manipulate individuals away from getting the vaccine. And obviously, whoever's tweeted this, they have the right to their opinion, but it's not really scientifically valid, is it? Because there's very good reason why the vaccine needs to be stored at -80 degrees, so at ultra low temperatures. As we've said, it's an RNA vaccine. RNA is less stable than protein, so it needs to be stored at a lower temperature than a normal vaccine. And the way this vaccine works is the RNA is taken up by cells, and then the cells translate the RNA into protein, and then this protein induces an immune response. So there are a lot of issues with this statement, and individuals who have a scientific education, who understand these principles, who understand what RNA vaccines are, would hopefully see that tweet and understand that it's misguided in many respects. However, there's no guarantee that people reading the tweet do have that understanding of the fundamental scientific principles. So again, just making the point that these education issues persist across borders and across cultures, and the best way we can do it, or we can deal with it, is to encourage education, provide the opportunities for individuals to become educated. There's this thin line that needs to be walked down between making people feel like they're looked down on because they don't understand scientific principles. That's something we really want to avoid. It's absolutely no one's fault if they don't have the same education level as someone else. Not everyone can have a degree in science, right? It just doesn't work like that. So what we should really be doing as scientists is providing opportunities for individuals to understand these fundamental scientific principles, rather than kind of shaming individuals for not understanding science, which is a really unhealthy approach. In the last few slides, I've discussed HIV and AIDS denialism, which is kind of another example of lack of education or lack of understanding of scientific principles causing people to become misguided. So HIV and AIDS denialism is the position that HIV doesn't cause AIDS, or AIDS is not deadly, or HIV doesn't exist. It's essentially the position of denying that HIV and AIDS is a valid health concern, and that the scientific consensus on HIV is true. So arguing that that is not the case, and arguing that, again, HIV doesn't cause AIDS, or AIDS isn't deadly. This is interesting because we have a quote from the former president of South Africa here. As I've said, we tend to wrap lack of education up with poverty, but that's absolutely not always the case. So we can have individuals who are very competent and very capable and accomplished within their field, but when it comes to science, they really have very little understanding, or they, they potentially lie. I mean, it's the real world. People lie. Individuals have ideologies. Politicians have agendas, and they have positions they want to hold, and therefore they could be misunderstanding science, or they could be deliberately misleading people. It's not really clear, and it's regardless. Occasionally you'll have politicians saying things that are unscientific, and that's what we have here. So it's essentially the President of South Africa taking the position that AIDS is not caused by HIV, so taking the position of HIV and AIDS denialism. And this is obviously super dangerous, because he's potentially influencing a lot of people. He has influence over the country. It's unfortunate, but it was seen quite a lot in Africa in the 80s and the 90s. And again, there's contextual things going on where it's not just a lack of education, it's also ideologies at play and politics. There was a position held for a while that the idea of HIV and AIDS was inherently racist, in that it was a kind of colonial invention to paint native Africans as being infected, and to kind of disparage the communities and the cultures, which we know scientifically that's not true. But it was a somewhat widespread belief in Africa for a period of time, hopefully less so now. And this paper outlines the fact that belief in these conspiracy theories, so these theories that are not founded in science and they're not empirically supported, are associated with non-adherence to antiretroviral medications. And this is in African American men, so this is in the USA. So obviously these aren't just kind of vague philosophical disagreements we're talking about. These are disagreements in the science, or not disagreements in the science, or disagreements with the science that impact individuals' willingness to engage with the therapeutic interventions, which is obviously a really significant deal. And finally, I just wanted to make the point that in many populations, recent immigrants are inordinately impacted by infectious diseases. The reason being that there are specific barriers that can prevent or disincline recent immigrants from engaging with healthcare services. And the most obvious would be language. So if an individual is a recent immigrant, potentially they don't have a solid grasp of the native language yet, and this can be a barrier to them receiving healthcare. Just navigating the community as a whole can be a barrier. So they don't know where to go, they don't know who to speak to, and these are all potential situations that recent immigrants can find themselves in. The lack of social structure and the lack of social support, if they're not a part of a local community yet, this can also impact their willingness and ability to engage in healthcare systems. Okay, so just some things to think about there, some pathways you can take your evaluation down. Hope you found it useful. I hope after this lecture you have a real solid understanding of not just the therapeutic interventions themselves, but also the way these therapeutics can be implemented in terms of the wider context, in terms of some comparisons across antivirals and vaccinations. So thanks again, and I'll see everyone Wednesday. Take care.